Today, it's my pleasure to talk with Dr. Steve Waxman. He's a professor of neurology and neuroscience at Yale University School of Medicine, where he's also the founder and director of the Center for Neuroscience and Regenerative Research. He's a neurologist and a PhD neuroscientist. In fact, he doesn't see patients now. He's been focusing on research throughout his career. He's an expert on pain, and in particular, the nerve cells that perceive pain in the periphery and all, feet, hands, wherever on our body, and how those pain sensing neurons function and communicate the pain signals up to the spinal cord and brain. And he's been discovering and characterizing genetic abnormalities that lead to pain syndromes in which the person may either have hypersensitivity to pain, or in some very rare cases, complete insensitivity to pain. Dr. Waxman, before we get into the science, could you give our listeners and viewers some background on you?
Sure. I suppose if you wanted a one-sentence summary, it's that I'm passionate about science, and I'm also passionate about family, our grandkids. But I was the first person in my family to go to college outside of Rutgers, Newark. I came from a family where neither of my parents went to college. I didn't have any scientists or doctors in my family, but from the earliest I can remember, I wanted to be a scientist. And I was fortunate in having a few teachers who not only encouraged me, but who pointed out that I could do well. I didn't know about high horizons, and they pointed them out. And so for me, science is the best job in the world.
It's fun.
I just love what I do.
New things every day, and you get to interact with a lot of really smart, energetic, young, trainees, and it's very, very rewarding.
It's very rewarding, and so you learn something new every day. Almost every day, a new puzzle gets thrown your way. Some of them you could solve, some you can't. As Mark said, as you said, Mark, I get to work with incredibly bright people who really share the same value systems. As a scientist, you get to travel, you get to meet really, really interesting people, and so, as I said, it's the perfect job for me. For me, being an MD PhD also is the right fit. I see patients in a research context, but what my MD does is it really provides a crash course in human biology. And so I'm able to listen to a discussion about a molecule, and I may know a little less about that molecule, but I often can have a sense of where it fits in the scheme of human biology, both normal human biology and diseased human biology.
Yeah, and as we'll see as we go forward in this, that it's not that common that someone starts out doing the basic neuroscience and then ends up, working towards treatments and actually doing clinical trials in patients. So we're going to learn about that in regards to pain from you in this talk. Now, your undergraduate, you went to Harvard, then your MD, PhD was at Albert Einstein College of Medicine, where you got interested in axons and these long fibers that are coming from the cell bodies of the neurons and going, say, from the spinal cord to the big toe. And do you want to briefly, before we get into pain, kind of talk about the evolution of your interest in axons?
Sure. Actually, my interest in axons, nerve fibers, dates back even earlier, because as an undergraduate at Harvard, I worked with J.D. Robertson, and he was best known for unraveling the bilayer structure of cell membranes, but also it was J.D. Robertson who discovered the spiral architecture of myelin sheaths, just a beautiful, beautiful piece of biological architecture. And so he was studying axons in that part of his lab. And with his encouragement, I took a semester as an undergraduate away and worked at University College London. I
Oh,
recall getting there and seeing the breadth of neuroscience. J.Z. Young was chair of the the Department of Anatomy and Cell Biology. He had discovered the squid giant axon, and he was one of my mentors. Sir Andrew Huxley was one floor up, and Sir Bernard Katz, a second Nobel Prize winner, was in the adjacent building. And John O'Keefe, who 30 years later got the Nobel Prize for place cells, was one floor upstairs. So it was scintillating. But a theme that I somehow got attracted to was nerve fibers, how they work, their overall structure, and their molecular structure. And that leads into the electrogenosome, the ion channels that shape communication, shape action potential, generation, and transmission along neurofibers. And then if you fast forward a few years, and in my first faculty job, I was an assistant professor at Harvard and MIT. And interestingly, my labs were in a building 36 at MIT was RLE, the Research Laboratory of Electronics. I was surrounded by computer engineers. This is where information theory was born, cybernetics. Norbert Wiener worked in that building, and the top floor was sensory neurobiology. And so there was immense interest in quantitation and communication. And as I moved through those initial years, I became more and more convinced that that surely if we could learn something from a squid axon, a squid giant axon, or the axon of a frog, then surely there were things we could learn about a human axon and even from a diseased human axon. And back in 1978, I edited a book. It was called Physiology and Pathobiology of Axons. And the subtext was to the axonal community, hey, we really need to take basic science and at a minimum view diseased human axons as a form of comparative physiology that can teach us, and maybe, just maybe, we could have an impact on disease.
yeah. And your first, well, I guess you can tell us. My understanding is your first work, disease-related work, was on studies relevant to multiple sclerosis, where at the time, the thinking was these insulating cells, the Schwann cells that wrap around and insulate the axon and enhance its conduction velocity, that they degenerate, and then there's reduced conduction velocity. But then it became, evidence started emerging that actually it's not just that, the axon, there's actually problems with the axon.
The axon is a gorgeous piece, just at least to me, a gorgeous piece of biological architecture, because the myelinated axon, in contrast to non-myelinated axons, which are sprinkled with sodium channels in low density throughout their course, the myelinated axon is highly differentiated. And so there is a very high density of sodium channels clustered just where you need them, around 1,000, 10 to the third channels per square micron at the node where you need them. And then in the internodal axon membrane, under the myelin, there's a very, very low density of sodium channels, so low that that part of the membrane can't sustain conduction. So that's a piece of biological architecture of basic interest. But then if one asks, well, what happens in multiple sclerosis, where myelin is sheared away, that opens up a whole new set of questions. What happens when you expose this membrane? You lose the resistance, you lose the capacitative shield. And the previous sodium channel poor membrane is still sodium channel poor. You're exposing membrane with nobody home. It can't generate action potentials. And what that taught us is that in addition to demyelination, it's the exposure of sodium channel poor membrane that causes conduction block in multiple sclerosis, in the brains and spinal cords of human beings with multiple sclerosis. But the next question is, when you go to medical school, you are taught, and we teach, that following damage to the brain and spinal cord, there's very little, if any, functional recovery.
Right.
That's one of the catechisms. But there's an exception to that, and the exception is multiple sclerosis, where we routinely see remissions. So somebody will develop optic neuritis, lose vision in an eye, and then 8 weeks later, with or without treatment, they will experience a remission and regain vision, or regain motor strength in their legs. What's happening there? And it turns out that the demyelinated axon membrane in some axons reorganizes and acquires a higher than normal sodium channel density, high enough to sustain impulse conduction through the demyelinated zone. Remyelination doesn't occur. And so what we learned is we now understand the molecular substrates for remission, recovery of conduction in demyelinated axons, which underlies recovery of function. And we, of course, want to emulate that process. We can't yet do that, but we're peeling away layer by layer in terms of the molecular neurobiology of axonal injury and dysfunction in MS.
And then when did you start working on pain? Because you know, for the last 20 years at least, right, you've been focused a lot on pain and sodium channels. So could you talk about what are sodium channels and why might they be important? Why are they important normally for pain? Pain isn't often a good thing, right? It alerts us that we've been injured. But on the other hand, when pain signals keep coming, even when there's not any obvious proximate cause, then you know, that's chronic pain.
Sure. I I actually worked on pain in 1969, 1970, when I made a second trip to University College London, UCL, and I worked with Patrick Wall. Pat really revolutionized pain research and brought it into the modern era. He was a remarkable mentor, and he had a small lab, much smaller than by today's standards. He usually had one postdoc, one technician, and one student, and that student got to work with him cheek by jowl, day by day. In any event, we worked on the barrage of impulses that is excited by acute axonal injury in peripheral nerves. That was around 1969, and the paper was published around 1973. Wall, Waxman, and Bassbaum. But I worked on other issues. I worked on spinal cord regeneration. I worked on MS, the molecular neurobiology of MS that you just heard about, until in around 1994, where we asked the question, what happens to the program for expression of sodium channels when you injure the axon of a neuron? And whereas Hodgkin and Huxley talked about the sodium channel, which was sometimes interpreted to mean that there might be one sodium channel, by 1994, the molecular revolution was in full force, and it had become clear that there are multiple sodium channels encoded by multiple genes in mammals, in humans, with slightly different amino acid sequences and with different physiological and pharmacological properties. So we looked at dorsal root ganglion neurons, and we said, what happens to their program of expression of sodium channels if you cut their axon by peripheral nerve axotomy? And we found something remarkable, which was what we called at that time the type 3 sodium channel. We now call it NAV1.3. normally produced during embryogenesis robustly, and then it turns off, and it is not present in adulthood until you cut the axon of DRG neurons, and then it is strongly re-expressed at the RNA level and the protein level. And we went on to show that NAV 1.3 has a number of biophysical properties which poise it to make neurons hyperexcitable. It operates to amplify small subthreshold depolarizations. It recovers remarkably quickly from inactivation. And so once it turns on and it's present in DRG neurons, it poises them to fire at lower than normal thresholds and in higher than normal frequencies. So that's when we began to really roll up our sleeves in terms of dissecting the contribution of ion channels to pain. And as that moved ahead, that work, we did the earliest studies on the profiling, the physiological profiling of NAV 1.7 sodium channels, then NAV 1.8. Slayman Dib-Hodge, my colleague and friend, discovered and cloned NAV 1.9, and so we built up our library of sodium channels. NAV 1.7, 1.8, and 1.9 are peripheral sodium channels. They are preferentially expressed in peripheral neurons, like sensory neurons. And this was very important because if you think about neuropathic pain, and it occurs commonly after nerve injury, in diabetic neuropathy, in chemotherapy-induced neuropathy, shingles neuropathy, post-herpetic neuralgia, it's a major unmet need. Neuropathic pain is not responsive or not very responsive at tolerable doses to many of the existing medicines. And yet, when patients with neuropathic pain go to the dentist, they receive a local injection of a local anesthetic, lidocaine, bupivacaine, one of the cane drugs. These are non-specific sodium channel blockers, but when they're locally injected into a peripheral nerve, there is no pain. And yet you could not take these drugs and turn them into systemic medications because they affect neurons in the CNS, they affect the heart, you get double vision, impaired balance, confusion, sleepiness. So that raised the question, might there be one or more peripheral sodium channels present in and important for the function of peripheral neurons, but not the brain and spinal cord? And NAV1, and it was a holy grail finding these. A number of labs all worked on this. We were one of them. NAV 1.7, NAV 1.8, and NAV 1.9, all in a broad brush way meet the criteria for being peripheral sodium channels. And so there we were working on NAV 1.7, 8, and 9. And
these three different sodium channels that are in the nociceptive neurons, the neurons that convey pain. So they all conduct sodium. So when the membranes depolarize, they open, sodium goes in. But my understanding is there's a difference in their certain aspects of their properties. Is that right?
Yeah. So here the differences in amino acid sequence endow the channels with different physiological properties. So NAV 1.7 has a property called slow closed state inactivation. And what that does is it prolongs the depolarization that's produced when it's activated. And that, together with its left shifted, hyperpolarization shifted voltage dependence, allow it to respond to slow, small, subtle depolarizations well below the threshold for spike initiation. So 1.7, NAV 1.7, is excited by these slow subthreshold depolarizations, and it amplifies them. And it acts in the subthreshold range to bring the cell to threshold where NAV 1.8 takes over and produces the rapid upstroke of the action potential. So the different channels work in concert as collaborators, but each of them does a specific and important thing.
And does NAV 1.9's properties differ from the other two?
Yeah, 1.9 is probably the most mysterious. It is inactivated at normal resting potential. You only see these channels when you hyperpolarize neurons. It also produces a subthreshold slow depolarization, precisely why you need NAV 1.9 plus 1.7 is not yet clear. They certainly are quite different molecularly, physiologically, and we're sorting out why you need both in some neurons in order to get the level of electrogenesis that we usually see.
And now might there be, you mentioned with the demyelination in multiple sclerosis, you get this recovery of function and the sodium channels are aggregating in the membrane. Might there be differences in the localization of the different channels? Maybe one's more in the ends of the axon, another one in whatever, or more in the cell body. When you do electrophysiology, you're mainly measuring what's happening in the cell body, right?
Yeah, Mark, you're talking about an important question. And in terms of DRG, dorsal root ganglion pain signaling neurons, this is an incredibly important question, and you've pointed out an important caveat to all our studies. We generally do our patch clamping on the cell body because that's where it's easiest to do it. And sort of an implicit extrapolation is that things might be like that along the axon, but we do not know that that's formally so. Dima Dmitro Vasilyev, who just arrived here 2 months ago from Kiev, is the only person in the world. We published a paper in 2012 recorded doing patch clamp from single DRG axons in culture. These are tiny axons, and it was there are a couple of microns in diameter. It was a heroic effort by Dima. We're continuing that work now. What we learned is that the axons have a resting potential very similar to that in the cell body. That was not previously known. And we were able to demonstrate quite clearly, because we could do voltage clamps in these axons, that they express NAV 1.7 and NAV 1.8, which act sequentially as they do in the cell body. But there are a bunch of unanswered questions. Are the channels in a peripheral axon distributed uniformly or at random, or are there clusters? Might there even be small clusters that support microsaltatory conduction at points of low safety factor? And an example is a branch point. Do you need a booster zone? Our computer simulations show that you can facilitate high frequency invasion of both branches at a branch point if you put in proximally a small island of increased sodium channel density. But we don't know that occurs in these tiny axons in vivo. We have a lot of work to do, and we're doing it.
And there's now molecular genetics and essentially genetic engineering approaches have been used extensively to generate animal models. For example, it's possible to knock out or inactivate, in this case, a particular sodium channel gene, or even put a specific mutation gene in an animal, and then I guess in the animals look at some, I guess relatively, they can't tell you, ouch, you can't say what's your pain on a scale from 1 to 10, and they'll tell you that you give them like a mild painful stimulus. So what happens if you remove the gene for NAV 1.7 from mice?
Well, so The mouse models are useful, but of limited value. They have produced a sometimes uh, confusing array of clinical pictures. We've taken mutations of NAV1.7 from patients with painful peripheral neuropathy. We've built mice with those mutations, and the mutations make their DRG neurons hyperexcitable, but we do not see overt signs of pain in those animals. There's a mismatch, and we continue to, along with many others, and we're not the only group that has seen that mismatch. The Clifford Wolf's group up at Harvard came to similar conclusions. And it may be that we don't interrogate our mice as well as would be optimal, but that's where we are right now. We've learned most from human genetic syndromes and iPSCs, iPSC-derived sensory neurons from humans. And in these humans, we can phenotype things exquisitely.
And could you briefly explain what an IPS cell is?
Sure, an iPSC, an IPS cell, is an induced pluripotent stem cell. So from anybody in the audience, we could take a sample of blood, or we could take fibroblasts from a skin biopsy, and we can dedifferentiate them into iPSCs, induced pluripotent stem cells. The regenerative medicine community is trying to use these cells for regenerative medicine. Can we induce those cells to redifferentiate into heart cells for repair of the injured heart, or pancreatic insulin secreting cells for diabetes, et cetera, et cetera? We're using those cells as disease models. We differentiate them into sensory neurons, and we're asking, okay, now we have sensory neurons derived from an iPSC from this particular patient with that particular patient's 23,000 genes, their entire genome. And that's been very, very instructive. I think most instructive is how we've used those cells, that stem cell model, to dissect pain resilience. But maybe before even getting there, the story of human genetic pain syndromes related to channelopathies may be of interest to people
Yeah,
listening.
So you've written a book. It's called Men on Fire, and
Yeah.
the hunt or the search for the pain gene. And could you go over this? Is a very fascinating story that unfolded beginning in Alabama and going through Beijing and you. So
Well, this is a story in molecular genetics. We're really interested in neuropathic pain due to dysfunction of the nervous system. And every neurologist sees a lot of neuropathic pain in their clinics. We see it all the time. There's not a lot we can do about it, but it's quite common. What we don't see is families with inherited neuropathic pain, and yet we wanted to find them if they existed. And so why search the world for families that may or may not exist? And the reason, a good example is the statin medications, the statin drugs. These are drugs that have revolutionized cardiovascular medicine, and they had their origin in the discovery and characterization of incredibly rare families where everybody was having heart attacks in their 20s. These families had inherited hypercholesterolemia, a tiny number of families, but once they were available and studied, that pointed the way to the mutant genes and to the target molecules, and opened up an entirely new chapter in cardiovascular medicine. So we surveil a population with colleagues of 3 billion people, looking for the rare experiments of nature, families with inherited neuropathic pain. The disease we study is a disease most physicians will never see but if they see it, they never, ever forget it because it's so striking. It's called the man on fire syndrome, inherited erythromelalgia, IEM. And these people feel searing, burning, scalding pain in response to mild warmth, wearing a pair of shoes, putting on a sweater, going outside on a day when it's 71 degrees Fahrenheit. They describe it as feeling as if a blowtorch had been used or there was hot lava within their bodies. And they will, it occurs in a neuropathic distribution, the hands and the feet, primarily the feet. These individuals will keep it, as I said, the attacks are triggered by warmth and they are relieved by cooling. And these people will keep their hands and feet on ice to the point of getting gangrene. There is, for most of these families, nothing works. There is no therapy at the moment. In the inherited form, these are patients who have gain of function mutations of NAV1.7. The mutations shift activation in a hyperpolarizing direction. A leftward shift of 6 or 7 millivolts, in some cases 12 or 13 or 14 millivolts. Our record holder is 18 millivolts. That's an immense shift in activation, making it easier to turn on the NAV1.7 channel, which is acting as a volume knob. It's acting as a volume knob on DRG neurons. And so what happens when a DRG neuron, a pain signaling neuron, is carrying these mutations? Its volume knob has been turned on, turned up, its threshold is down, it's firing at a lower threshold than it should, and it's firing at a much higher frequency than it should. And it's sending the message into the spinal cord when the message should be bop, bop, meaning I'm slightly warm. It's going bop, 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 saying I'm on fire. This is a really graphic example of a gain of function mutation. It's been a remarkable story in a human sense because the story did bounce around, as Mark said. The first families were found in Beijing, China. The point mutations were found in two families there, but that initial paper did nothing to address the issue of causation. In order to show that a mutation causes a disease, you need to study the mutant channel by appropriate voltage clamp, and then hopefully put the mutant channel in the cells where it normally resides, and show that the mutation perturbs the behavior of those cells. And the initial discoverers of the first two cases in Beijing had not done any of that. They were brilliant geneticists, brilliant medical scientists. They were not channel biologists. They were not neuroscientists. And so that ignited us to move ahead. We have a large family in Alabama. It's been exquisitely well studied. It's 6 generations, 60 individuals, many of them debilitated by this disease. We now have a library of families, kindreds from around the world. It is interesting, at a time when large GWAS studies of cohorts of tens of thousands of patients are viewed as the benchmark, we feel, and those just do the statistics. They don't look at mechanism. They don't do anything in a reductionist mechanistic sense, but they find associations. We've tended to look in a very reductionist mechanistic way, and we've learned a lot from small families. And in fact, I wrote a commentary in the journal Pain a couple of years ago called Channelopathies, Big Lessons from Small Kindreds. And these kindreds have taught us a lot. It's been a very rich experience because these individuals have given us an immense gift. They give us their DNA. They share intimate details of their medical histories, their family histories, and they look to us for advice. And it really, and when they look to us for advice, I'm very candid, and I tell them, I know you want a therapy for your loved one. We don't yet have it. And not only do we not yet have it, it's challenging work, and there is not going to be a cure tomorrow or next month or in a year or maybe even in a few years, but we're making progress every month. We're learning more and more, and your channel, your mutations have shown us that NAV 1.7 is a master controller of pain, and we are getting there. And in my view, it's highly likely that we will have new and more effective therapies for pain.
And is a lot of neurological disorders that are inherited in a similar manner we call autosomal dominant, where if either your mother or father has the gene mutation, then you will inherit. You have a 50% chance of having the mutation. And a lot of, I work a lot on Alzheimer's, Parkinson's, other neurodegenerative disorders, and many of those there's always an age of onset, that is, little kids don't have symptoms, but then as they get older, they'll develop symptoms. Is that true with these inherited uh, hypersensitivity to pain, or is it?
Yeah, sure. So inherited erythromelalgia is autosomal dominant. If a parent has the disease, the child has a 50% chance of inheriting the mutated gene. And if 100% penetrance, you've got that mutation, you've got the disease with a very strong phenotype beginning in infancy.
Okay.
Now, there's another disorder, and that's channelopathy associated insensitivity to pain. These are families who have biallelic loss of function mutations for 1.7 on both chromosomes. So these patients do not make any NAV 1.7. And these patients have a remarkable picture of total insensitivity to pain. It's a stunning degree of insensitivity to pain. Painless fractures, painless burns, painless childbirth, painless dental extractions. And so we've got both sides of the coin. Strong gain of function mutations, horrific pain. You don't make the channel, total loss of expression of that channel, total insensitivity to pain. And that's a very strong degree of genetic validation that ignited a storm of interest in the biopharma industry. We have asked, all these are incredibly rare syndromes. They're model diseases. They teach us a lot, but they're incredibly rare. And so when I lecture on this, after introducing these two syndromes, I often show a slide saying something like, "What about the rest of us?" And in that slide, I lead up to neuromas, and those are bundles of injured nerve fibers after peripheral nerve injury. And there we see abnormal accumulations of NAV 1.7, which is contributing to the pain associated with neuroma. That's neuropathic pain after nerve injury. And together with colleagues in the Netherlands, Karen Faber, Ingemar Merkes, and their team, we've been studying patients with small fiber neuropathy. That's a peripheral neuropathy that preferentially affects the small A delta and C fibers. And 5% of those patients carry gain of function mutations of NAV 1.7, but much more subtle. The shift in activation is much smaller than for the IEM mutations, or there may be very minor impairments of inactivation. But these mutations do make DRG neurons, pain signaling neurons, hyperactive. That raises the question that Mark asked, and it's a really important one. These patients are carrying these mutations throughout life. and yet they develop the signs and symptoms of peripheral neuropathy in adulthood. That's when most cases of peripheral neuropathy develop. 60 years old. So what's different, since you carried the mutation for your life, what's different when you're 60 compared to when you were 40? There's a time dependence that we don't understand. We're working on it, but it's an important puzzle. And there's also a length dependence, because peripheral neuropathy affects the longest fibers first. And so it affects the feet and then the hands, the longest nerve fibers. Why the length dependence? The original traditional view was that it had to do with axoplasmic transport and the longest fibers being most susceptible to any impairment of transport of trophic stuff. That's how old the hypothesis was. We've raised the question that there may be other things going on. We know that energetics plays a role, at least in culture, in injuring nerve fibers carrying these mutations. And we suspect that there may be age dependent and length dependent abnormalities in mitochondria and energetics that contribute to the age dependence and length dependence. But that's work that's going on. And again, to the younger people in the audience, the fact there's still so many unanswered questions is part of the fun of being a scientist.
So the mitochondria might fit right in with the sodium channels, because mitochondria produce ATP, and one of the main utilizers of ATP out in axons is the sodium pump, which we haven't talked about, but you get this sodium influx after the neuron fires, and then it's important that the sodium be removed from inside the cell quickly so that the membrane potential is restored. And so there are these pumps, sodium pump, that pumps it out. And it is known that during aging in general, the function of mitochondria is compromised, and you get, in a lot of cells, we've studied this some in relation to Alzheimer's, neurons in the brain, accumulation of dysfunctional mitochondria. And so, yeah, I mean, that wouldn't, so hypothetically, if you did some manipulation that caused, say, a 20% reduction in the sodium pump activity, would that lead to pain hypersensitivity because the cell isn't restoring the sodium gradient quick enough?
Mark, you're talking about something really important. Some of our readers may know Peter Stice. He currently is a professor at University of Calgary. Peter came as a research fellow in, I think, 1989 or 90, and did absolutely stunning work. Journal of Neuroscience, 1990, Stice, Ransom, and Waxman, and he showed in axons within the brain precisely the kind of interaction between sodium channel activity and ATP and energetics that you're talking about. And in fact, Peter showed that you could protect axons from a variety of insults by blocking sodium channels with TTX or with clinically used sodium channel blockers like carbamazepine and phenytoin. That theme is continuing. He did his work in white matter of the brain. We're continuing that in our models of peripheral nerve.
That's great. Okay, so you've talked about a few drugs in the last few sentences there that block sodium channels. What's going on in terms of drug development? You pointed out that it would be really valuable to have a drug that specifically blocks NAV 1.7, or perhaps another sodium channel that's only in the peripheral neurons. What's going on there as far as moving it to clinic?
So yeah, so NAV 1.7 works in tandem with NAV 1.8, and we would predict that a subtype selective blocker of each of those would leave the other channels alone and have a favorable side effect profile, but would attenuate pain. and work is going ahead on both fronts. And you could imagine blocking the channel with a small molecule drug, or with a peptide, or with an antibody, or by a gene therapy approach. And all of these are being explored. Um, uh, when I talk to, when I give my lecture, I talk about the fact that translation, in my view, is achievable. It's been talked about as as the valley of death, going from the laboratory to the clinic. In my view, it's achievable. But I also point out that if you want to get involved in translation, you need to buckle your seatbelt and anticipate a long ride and sometimes a bumpy ride. We began.
Fear no evil as you, whatever, pass through the valley of death.
Yeah, fear no evil. So we began working in 2007 with Pfizer. They had a NAV 1.7 subtype specific blocker. It was their PF771 compound. And so the idea was to study their blocker in our IEM patients in a study that we had designed.
Yeah.
And we began working with them in 2007. That's when Yale signed the agreement. The paper was not not published till 2016. This is a long road, but in a word, they had their 771 blocker. We did deep phenotyping on patients with IEM from throughout North America. 13 patients, that's all we could find in North America. And of those 13, 4 qualified for a clinical study with their blocker and agreed to participate and to give us IPS cells. And so in those 4 patients, we made iPSCs. We now had iPSC sensory neurons from the IEM patients, from normal controls, and the IEM sensory neurons were much more excitable. They were grossly hyperexcitable. So we had a model of disease, and then we studied the effect of the Pfizer 771 1.7 blocker. And it reduced the hyperexcitability in 3 of the 4 lines of iPSCs, but not the 4th one. And then we did the clinical study. This was double-blinded and placebo-controlled. Each patient received 2 doses of the drug, 2 doses of placebo, crossover, very, very careful design. The patients whose iPSCs did not respond, that patient didn't respond. Of the patients whose iPSCs did respond by becoming less hyperexcitable, all 3 of them responded robustly to the drug on at least one of the 2 exposures, and 2 of the 3 responded on both of the exposures. So the good news is that this study reinforced the notion that iPSC-derived neurons could be used as a disease model for drug testing, for preclinical testing of drugs. And this was the first time we were able to show, that a 1.7 blocker could reduce pain in humans. And that's an important step forward. The cautionary note is that this was 4 patients. It took a long time. It took the resources of a Pfizer. We couldn't have done it alone. But to me, it's very encouraging. I should tell you also, we have another family with IEM, and on the basis of our modeling, we predicted that their mutation not only would produce IEM by shifting activation, but also would sensitize their channels to carbamazepine, a clinically used drug, Tegretol. And on that basis, we did a clinical study in those patients, and we found that in both of those patients, double-blinded study, carbamazepine markedly reduced time in pain, length of pain attacks, number of awakenings from pain, etc. So again, a very encouraging result, but again, a small N requiring a lot of effort.
Yeah. Although, my understanding is there, for very rare genetic disorders like this, the the bar that you've got to hurdle over with regards to FDA approval is a little lower. Is that right? Than something that you would just be giving to the general population, or whether it's some common disease.
I'm not an expert on regulatory affairs or FDA criteria, but that's my general understanding.
Yeah.
And for me, the important thing is, number one, there are these cohorts of small numbers of patients. The N is small, but every life is precious, and the quality of every life is precious.
Yeah.
And if we can deliver pain relief to them, I will go to sleep very satisfied. And the second point is, for me, these are proof of concept studies.
Yeah.
And we learn from every patient, we learn from every disorder. So my hope is that we're learning or reinforcing the notion that NAV 1.7 plays this important master switch volume knob role, and is an opportune target, or its partner, NAV 1.8.
And are there any other targets that, there's more channel proteins that affect excitability, potassium channels. There's these proteins called trip channels. There was a Nobel Prize given for work on those earlier. Are any of these people pursuing these?
Yeah, we've been studying a disorder called trigeminal neuralgia. It's an episode where you get severe paroxysms, attacks of facial pain, like an axe hitting you in the face. And most cases are sporadic, but there are a small number of families. We've been studying those families, and we have found 2 mutations of trip channels. And one of those mutations produces hyperexcitability of the relevant sensory neurons to the face by producing what's called an omega current. It basically produces a new pore through the channel. So that's one thing we're working on. But in addition, what I'm really interested in is, in our families with IEM, the man on fire syndrome, we usually see excruciating pain in all the affected people. We have 2 families, which are outlier families, and each of these families has one individual whose pain is not so bad. They carry the mutation, and their pain is nothing to write home about. And so these are pain resilient people, and we desperately want to know what they have to teach us. And so we've phenotyped them, we follow them, we do pain diaries for 14 weeks, and we establish that indeed they are truly clinically pain resilient. And the question is why? So what we've done is to make iPSCs from these little pain patients with IEM and their bad pain relatives. And so now we have the sensory neurons from both, and the bad pain iPSCs are going bop, 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 bop. And the relative with the same gain of function NAV 1.7 mutation and little pain has hyperactive DRG neurons, but much less hyperactive. Instead of bop, 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 the neurons are going bop, 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 bop. So we have disease in a dish, but we also have captured inter-individual differences in severity of the disease. And the next question is why? So what we've done is to do whole exome sequencing, all 23,000 genes. That's become pretty standard. And then we've asked, okay, which genes are expressed in the electrogenosome of the DRG neurons? That is, which genes are related to firing of these cells? And what we've found in these pain resilient people is that they carry gain of function mutations of the KV7.2 and KV7.3 channel. Now, KV 7.2 and KV 7.3 act to produce something called the M current. It's a current that stabilizes neurons. And and we've been able to show definitively that these gain of function mutations are protecting those neurons from hyperexcitability. So what this does for us is enlarge our library of targets. And now KV 7.2, KV 7.3 are targets, and there we are. And that just feeds into our overall strategy, as you said, Mark, of there are lots of potential molecular targets. We want to take multiple shots on goal. And that's one of the reasons I'm confident that sooner or later, I don't think it's fair to say there may be. I use my words conservatively, but I think I can say reasonably confidently that sooner or later there will be new and more effective non-addictive treatments for neuropathic pain.
Yeah. There's one other class of interesting molecules. When I was at University of Kentucky, there was a couple, Kathy Albers and her husband, they worked on peripheral neurons and NGF, and they'd made this mouse that has, so if you completely knock out the NGF gene, the animals will die early in development. But if you then express the gene only in the skin, then they'll live. And so what's happened, and then I think, after that, it was later shown that NGF plays an important role in the sensory neuron fiber growth and actually in pain. And then we're working on another neurotrophic factor, BDNF, and there's clinical trials these people were doing in ALS, and they had to stop the trials because the patients were developing a lot of pain, like this spontaneous pain. So the point is that there are these neurotrophic factors that actually promote the growth of these sensory fibers, and they may influence pain. Is there any work going on in trying to target those for pain?
There is work going on. Number one, you're alluding to the fact that in vivo, nerve fibers are not growing in the dish. And so they're terminating in peripheral targets within skin and elsewhere. Keratinocytes within the skin are complex cells. There's an interaction with neurons that's being dissected right now. And in addition to the electrogenosome, there are lots of other molecules. There's the energetic cascade. There's the ensemble, and it's a rich ensemble of trophic factors. And various labs are looking at all of them, and I think they should be looked at. I've got a fairly large lab. I've got 35 scientists in my lab here at Yale, and despite that, we always are having to prioritize what's important and to strategize. It's one of the fun things going on, but I do think a challenge for us, the challenge as I see it, is not can we identify targets, it's can we prioritize the targets so we focus our efforts on the most likely to be to yield fruit targets. And that's an overriding thing in biomedicine overall. But there's no question this is immensely complex. And hopefully our reductionist approach is providing some windows of insight.
Now, this pain resilience is interesting. And, in the case where you made the iPS cells from patients who had a mutation but weren't experiencing a lot of pain, whereas their relatives with the mutation were, and you see an alteration that causes I would call a neuron intrinsic mechanism. I saw something, I think maybe it was in a talk you gave on developing biomarkers for pain. In the clinic, we have this scale, the kind of a uniform scale. What's your pain on level of 1 to 10. And of course, they're asking you when you're there, when you're in the clinic. And then they may ask you, I guess, what is it under certain conditions at home when it's, you mentioned hot outside with the IEM patients and so on, their pain goes up. And there's some effort, of course, it's your brain that's perceiving the pain, right? And kind of putting the emotional context onto the pain. And there's been some work looking at responses of neural networks in response to a peripheral pain stimulus. Can you say anything about that with regards to developing a biomarker of pain that it's a little, it doesn't depend on the subjective experience of the patient themselves?
So as an advocate for people with neurological diseases and as an advocate for people with chronic pain, it's essential that we get the biopharma sector to really partner with us and move ahead. And I can tell you that there are people making some strategic decisions in biopharma to steer away from pain or even to steer away from neurologic diseases in general, because it's a difficult area. And I think we really need to address that, both by suasion, but also by making pain research as good as it can be. So think about taking a drug into the clinic. How do you test it? As Mark said, at the moment there is no validated biomarker. You ask a patient, how bad is your pain? From 0, no pain, to 10, the worst pain you could imagine. That's a very noisy scale that's skewed by psychological state, sleep state, expectancy, et cetera, et cetera. The placebo effect is large, and that confounds results, and the animal models are not highly predictive of a human therapeutic response. And I can tell you that there are decision makers in some industrial biopharma companies who've said, quote, you would have to be crazy to attack that disease. And I just disagree with that. I think we have to go ahead. So how do we develop biomarkers? And one way would be to develop functional brain imaging. That work is going on around the world. There are patterns of brain imaging that are associated with chronic pain. They're not one-to-one biomarkers for chronic pain, but they're associated. And so when we did our study of carbamazepine in our patients with IEM, we did brain imaging. And what we found when we triggered their pain with heat is that they had an abnormal pattern of brain activation of the nucleus accumbens and the anterior cingulate. And these are areas of the brain that are associated with chronic pain. They're associated in general with reward and punishment. If you go to the casinos and lose a lot of money, these areas light up. But in any event, that's what these patients showed when they were initially studied. When we treated them with carbamazepine and their pain improved very substantially, their pattern of brain activity reverted to a normal pattern of activation of parietal attention and parietal somatosensory cortex. And that was interesting to me because it said not only did these patients had had their IEM for decades, and they had the abnormal pattern of brain activation. And here we saw that after a few weeks, it reverted to normal. It was not locked in. It was not hardwired. And that's an optimistic note. But in terms of the methodological question, is can that type of thing be turned into a biomarker that will have predictive value and be useful in moving toward the clinic? It's a very, very important area, and stay tuned.
Yeah. All right, we covered a lot here, and I think people should take away from this, your optimism and, based on the science and the neuroscience of pain, the sodium channels, the progress you've made, and that and the development of new ways, the iPS cells, the development of cell culture-based screens for drugs that could potentially, all these methodologies, these technologies that have been developed over decades are kind of converging. And I think it seems to me, from what I'm getting from you, that the pace as far as identifying treatments is accelerating. The pace is accelerating, and maybe a while still, maybe a number of years down the roads. But it's, I would agree. I think it's very likely that this is going to lead to, and as far as the drug companies, I don't know, if I was the head of a drug company, I'd say, Dr. Waxman told me that chronic pain is more costly in terms of human suffering and dollars than is cancer and, what do you say, cardiovascular disease combined or something like that. Okay. So there's a huge number of patients. So just from the standpoint of like simple-minded thinking, if you could come, and in the case of pain, it's much easier than Alzheimer's disease, which is something I've worked on. And I can understand from Alzheimer's perspective, maybe, that by the time someone's symptomatic, there's always, neurons are gone. It's not like you can give them carbamazepine and all of a sudden, restore something, give them another drug and all of a sudden the person's perfectly normal, their learning and memory is fine. So I think pain is would be a really good investment.
You said you would mention that to a biopharma president. You would make a good biopharma president. We need to have decision makers thinking like you're thinking.
Okay, Steve. Well, I enjoyed talking to you and learning about sodium channels pain and the way genetics, biophysics, and cell molecular biology have converged over the years, and it must be very rewarding for you to see your work, starting way back. You said 1969, over in London, lead to now, at the end of your talk, the initial small pilot clinical trials, which are pointing in the right direction. So thanks a lot.
Thank you. Thanks, everybody.
All right. Goodbye.